Okay. So at this point, we have a multi-sample call sets of germline short variants, and we have lovingly filtered them at the variant level, meaning we have made some determination, some classification for each variant call that we had in our call sets, of how confident we are that there is really some variation and at least one or more samples in our call sets. So we have some reasonable confidence about the ones that we kept after filtering. Now we keep all variants in the output file by default, but all the others, all the ones that are considered not potentially artifactual are, oh, is it on a, stop. Stop. What are we doing? Is it on a show thing? All right. That's interesting. Oh, it was slideshow. Wrong combination of keys, apparently. All right. Sorry about that. So yeah, we've determined which variants we think are real and annotated that. But that's at the variant level. We still need to figure out at the level of each sample, we've assigned some genotypes for each sample. And now we want to go in and look at that and we'll do it primarily for the variants we're interested in that have not been filtered out. We want to do this genotype refinement step because we care about genotypes in a lot of use cases, not all use cases, but for many use cases, we actually care about having an accurate genotype. It's going to make a difference to us if a particular person is heterozygous or homozygous variant, what exact alleles they carry. And that can come into play in different contexts, in different ways. For example, if we're doing clinical genetics, we may need to know do my patients have 2 copies of a loss of function mutation? LOF stands for loss of function, and that's basically when a particular variant causes a problem that disables it, where you lose the gene function. If you're heterozygous and you have only one copy of the loss of function mutation, you may still have a functional allele of your gene, and so you may be less affected by whatever problem is caused by losing function in this gene. But if you have both copies affected, you may have much more severe problems. So that matters. If you're looking at, sometimes you have the case, difficult case of having parents with a child with a genetic disease, and the parents want to know what is their exact genotype, to find out what is their chance that if I have another child, the next child will also be affected. And then based on that, they can make family planning decisions. If we're looking at population genetics, we need to know what the genotypes are for association studies. For example, we'll ask, how does the number of copies of an allele affect the genotype? Which ties back to that first question, but in a more general population sense, instead of patient-specific. So we care about genotypes. Hopefully, I don't need to tell you more on that idea. So let's make sure we agree on what this means. So the variance call itself is just at the high level across a population of 1 or more samples. What are all the alleles that have been identified at the sites across the population, and what are kind of the parameters of this variance? The genotype is for one specific person exactly what alleles they have. So for humans, for diploids. So for example, this person is homozygous variant T/T, meaning both copies of their gene is mutated. So that's what we're looking at. This, at this point, we decided it's all set. We're not going to change it anymore. But here, we may still have reason to change some of that information because we have sources of information that we have not yet utilized in our analysis. And we want to make sure that we deal with these problems. So why might we have some uncertainty around our genotype assignments? In some cases, they're just not very good data at sites. There might be some artifacts that make it more complicated. And we saw that problem this morning with the uncertainty around whether our individuals had a GTT or GTTT allele at the site that they're mutated at. So there can be an ambiguity. In some cases, the genotype we assigned is actually wrong. And we're able to some extent, refine that, correct that post hoc. We're going to have to make some assumptions, but I'll show you the rationale behind those assumptions. And so we're going to use population data, for example, data that we have about the frequency of certain alleles in certain population. And that's something where you'll want to make sure that you're matching the person you're looking at to the right population, to the population backgrounds they're representative of, which is something that you can infer. Well, we won't cover the inference methods, but it is something that you can find out about your patient, your participants. You can use pedigree information if you have sequencing data from the rest of the family. That is very useful. And we're going to be able to use both of these as a source of information for calculating what we call the posterior genotype probabilities. And that just means we're going to update the probabilities we assign to the genotypes based on this information. And the idea behind this, and it is again Bayesian statistics, the idea behind this is, for example, if you ask the question, you see your coworker walk into the office in the morning carrying an umbrella. What is the probability that it's raining outside? And you have, as information, you have the observation that they're carrying the umbrella and a certain probability of rain. And what we're doing in calculating the posterior probability is taking into account not just the observation that we make, but also the prior. There's a prior here that we can apply, which is our prior expectation. And that is something that is going to be based, that's where that population frequency and pedigree is going to come in. Because if you're living in, let's say in London, and somebody walks in with an umbrella, and you know it rains a lot in London, you're going to say, yeah, I've got a very high prior, very high expectation that it's raining. However, if you're living in Los Angeles, California, where it doesn't rain very often at all, then it's a lot likely your prior is much lower. And the result, if you take that into account, is that you won't believe as much the idea that it's raining, the hypothesis that it's raining. So that's basically how it works. And we're going to do the same thing with our genotype assignments, where if we have, for example, a homozygous variant call for a variant where the allele, that particular allele is very rare in the population, we're going to be more skeptical that we've actually found a person who's homozygous variants, because it's just less likely. We just don't expect it to happen as much. We're not going to let that override the actual data that we have, but if there's some ambiguity, we're going to use that expectation to modulate our decision. That is the entire concept behind genotype refinement. Any questions at this point? Okay. So the workflow is that we have our filtered variants, traditionally recalibrated, but you might have neural networked them, deep learned them, I don't know how we're gonna say that now. Anyway, so you have your filtered variants, and you're going to apply population priors, you usually have that available. Family priors, if you have them, that is not always available. And you're going to feed that into a tool called Calculate Genotype of Series, which you might guess from the name does what? It calculates genotype posteriors. We like very straightforward tool names in GATK. None of that fancy calling tools based on, I don't know, hats and types of clothing, not to mention particular bioinformatics tools. But we like to have a tool name that says kind of what it does on the tin. Anyway, so that tool is going to give us posterior qualities updated in our variant calls, in the genotype calls specifically. And after that, we'll have some variant filtration and annotation steps that I'll go over at that time. Really interesting part is the calculating the genotype posteriors. And I'm going to show you, and so that's the command line, you feed it the pedigree if you have it, you feed it the population supporting alleles if you have it. So I'm going to show you a couple of cases where running that actually made a difference, and kind of walk you through the logic one more time with the example. So in this case, and I know it's a little bit busy, but basically it's a IGV screenshot, which you'll recognize by now. At the top, you have the original VCF showing a homozygous variant call, and we have the original likelihood of that genotype right here. So if you remember, the 0 is the one that was assigned, so that homozygous variant. And the second most likely, which was the heterozygous case, you can see is just a 3. The difference between 3 and 0 is 3, right? Which is a very small number. And that tells you that we were very unsure which way to go. We leaned a little bit more towards the homozygous variant, but there was a lot of ambiguity at this site. And in fact, if you look at the reads, you see this here, there's a majority of the data that carries the allele, but there are some reads that do not. And so you're like, well, what is it really? Right, and so we run the tool with here we have, I know you can't read it, but it's basically the population frequencies applied. And we have a very low prior for that allele because we know from looking at the relevant population that this is an allele that's quite rare in the population. And so when we apply the tools with that prior, we're going to reevaluate that judgment and decide, based on the rarity of this allele, it is a lot more likely that we actually have a heterozygous case that was just misidentified. And so we're actually going to change the genotype assignments for this call, for this person, and recalculate the probabilities. And so now we've corrected the genotype and increased our confidence in the genotype assignment. Any questions about this one? Yeah. I'm sorry, say that again? Yes. Yeah. So it's Fred scaled, normalized, yeah. Okay. So second case is kind of the opposite. We start with a heterozygous call right here. And here's your data. It looks like almost all of the reads have this allele, yet we have a heads call. And if you look at those PLs, the Fred scale likelihoods, we have 0 and 0. Now, originally, there's decimal places, and we round them out. And so that's how you end up with 0, 0. It's basically a coin flip. Like, there must have been a tiny, tiny difference that made the calculation go one way rather than the other, but it's so close that it's impossible to have any kind of confidence in that genotype. Like as it is right now, it's unusable. But we're going to apply population frequencies, and we know that this is a variant with that particular allele is really very common in the population. So it's not at all surprising that you might have a homozygous variance individual. So we apply that reasoning and that assumption. We then decide we correct the genotype. It is more likely that it's actually a homozygous variance case, and we reassign this confidence. Now, you realize that Q16 is still not huge. That's still not a very high confidence. And so we're still reflecting the fact that there was some original uncertainty, and it's not like we're super sure. It's just that this seems more likely. Right, that's an important distinction to make. And I keep bumping into this thing. Okay, any questions about this one? Okay, so we did some validations just to make sure that we weren't just, using circular logic to convince ourselves. And so we looked at some truth data and looked at how the calls were affected by where we knew exactly what the call should be from orthogonal evaluations. And we try to figure out what is the effect of applying these tools. Overall, the general effects we saw was that we often had homozygous reference calls that were underconfident, where we actually could have been more confident that these were correct. And so by doing this, we boosted our confidence in those correct calls. And this reflects some of the biases of the caller, some of the weaknesses of the caller when it does the genotyping. And so here we're compensating for these weaknesses post hoc. We also find that often the homozygous variant calls were overly confident. We were a little too confident that we were right in that case. But we're able to increase, to adjust our confidence to better reflect the ambiguity. So sometimes, now I showed you cases where we fix the genotype, we increase our confidence. Sometimes what happens is actually you decrease the confidence in a particular call. And that's okay. Like our goal is not to be more confident of everything. It's more to be accurately confident of what we have. But that's the most important part. Right, so those were the kind of the two main observations we had. We also looked in terms of, and this is a kind of a looking more closely at one of these. We wanted to make sure that if there was ambiguity, we didn't hit ourselves into being more confident than we should be. And we found that basically what happened was if there were calls where we just didn't have good data in the sequence and we didn't have good supporting information or accurately helpful information from other sources, those confidences tended to stay the same. So we tended not to touch them. So we weren't damaging our levels of confidence. But where we had useful information that actually helped, there we increased our confidence. And the nice thing with that is that some of these calls would not have been usable in downstream analysis, but now that we increase our confidence and we're like, okay, we're more sure that this is usable, we're able to rescue some of these genotype assignments. Does that make sense? Okay, all right. Now, there's another consequence of doing this is that we can evaluate how confident we are in the situation when we have a family pedigree, and we can use that information. We use the fundamental assumptions that we get from Mendelian genetics about inheritance. to evaluate our genotype assignments when we have the family pedigree. These are contingency tables that just map out all the possible combinations of genotypes that you might have when you have a trio, so a child with a mother and a father. And these are all the possible combinations. And we're going to use kind of this enumeration of possible combinations to, and again, I don't want to get bogged down in the math, but the idea is to calculate what's possible versus what's a Mendelian violation. And there are some cases where you need to have like the most extreme case where you have a homozygous variant child and two homozygous reference parents, like you would need to have two de novo mutations to arrive at that genotype biologically, which is quite unlikely. But you can track the probability of that event. And so for each of the combinations that I showed in the contingency tables, you can kind of track how likely that is to happen depending on the starting configurations. And you can plug in the actual genotype likelihoods that you got for each of the members of the family. And when you combine all that information together, that allows you to say for any given, oh, I guess this is just this. I thought I had another slide about this. It will allow you to assess for each combination how likely that is to happen. And so we use that information in the same way that we use population frequencies to evaluate and potentially adjust the probabilities that we have for the family genotypes for the child. Once we have that, once we've adjusted all our genotype qualities based on that external information, we're going to want to exclude some of them where, and we use as a standard default thresholds a GQ of 20. So if we have a genotype quality less than 20, that's to us that's kind of the threshold of usability. Anything below that, we're not sure enough to be able to use it. And so this is just a step of annotating those genotypes with a filter that tells us that we can use later in downstream analysis to ignore those particular variants because they're not informative. We can also annotate based on all the information we've collected and annotated. We can annotate which variants are possible de novos. And that gets back to the Mendelian inheritance question of saying, well, depending on the configuration of genotypes that we get after this refinement, which ones are still in a state of Mendelian violation where we say, like, this is not biologically possible unless you have a new mutation that has arisen in the child. So we want to annotate that. And as a reminder, this is when you have a child that shows up with variation that is not present in either parent. Now there's a lot of things that show up. There can be a lot of variants and genotypes that show up that are as possible de novos when you do a regular analysis, and that's because some of those are errors. To identify which ones are most likely to be true, there's a few criteria that we apply. We expect that, well, by definition, the child is the only one in the family trio who has the variant allele. We impose that it's something where that allele has to be rare, because if you have a common allele, it's more likely to have been inherited through some other route, as opposed to being something that arose de novo in the child. And one case figure that you can have, and this is something that has been observed, is if you have a parent who has a mosaicism, where they get sequenced and you can't pull out the variant from them. The genotype is homozygous reference, but some of their tissues are mosaic and actually have some variation. They can pass that variation onto the child in a cryptic way, and the child will express that variation. And then when you look at the trio genotypes, they don't make sense. But then if you go back and do some deeper sequencing on the parents, you find out that they actually do carry the variant. There's also the case where the data was just not good and there was an error that was made and they were misidentified. The mosaicism quite is interesting because it's a case of biologically induced error mode. But you do expect it to be a rare allele. And you're only going to believe a de novo call if the genotype qualities of the parents' genotypes are good enough, because otherwise it might just be that you have a parent who has the variant and you just couldn't tell. And so the genotype quality improvement tools help a lot in allowing you to get the right information to make those judgments. And this is an example of a clinical case. We touched on it briefly yesterday in one of the intros, if you remember. And this was a real case where one of the developers on the team consulted on a clinical case where they figured that the child probably had some kind of de novo mutation based on the disease they were exhibiting. And the original analysis of the trio sequencing, I believe it was exome sequencing, showed up 417 de novo calls. Now that's not something that you can go back to the lab and say, please verify in the lab and resequence or retest all of these sites. Just too much. It would be too costly. And so the first step of genotype refinement, based on the posterior, like adjusted genotypes, narrowed that down dramatically to just 17 candidates, de novo mutations. And after filtering based on the genotype quality, they brought that down to 8 very high confidence de novo candidates. That they sent back to the lab, they reanalyzed and did some resequencing on just those sites, just very cheap at that point. And they identified correctly, they ultimately identified correctly what was the mutation that was causative in that child. So that's helpful. And that's what that's for. You can tune the priors without going too far into that. We are relying, one of the components that comes into play is the frequency, the expectation of mutation rates that might cause de novo mutations to arise. And we use, there's kind of a built-in default that corresponds to the expected normal mutation rate in humans. But if you're working with either a population that's been highly exposed to mutagens, like if you look at Chernobyl area populations, you may have some assumptions that they might have higher mutation rates for whatever reason. Or if you're working with a non-human population that you're exposing to mutagens or things like that, you can tweak the model to adjust your prior accordingly, your prior of mutations. Okay. And so at that point, you have variant calls with high quality genotypes. The next steps will be functional annotation. So that's the prediction of the functional effects that I mentioned yesterday, I believe. We used to recommend a tool called SNP-F, but now GATK has a functional annotation tool that's called Funcitator. We're not going to cover it in detail in this workshop, but if you need to do any functional annotation, which probably will, Funcitator is the go-to tool now. It's a very new tool in the sense that it came out of beta development in January of this year. It was released into production-worthy status in January with version 4.1. And there's a blog post on the GATK blog that kind of gives you an overview of what it does and lists resources that you can use to help to use it. And that's my talk. So at this point, we filtered our variants, find genotypes, and touch briefly on functional annotation. The last thing we need to do is just the Callset evaluation. This is a very complex topic, and what we're going to do is give you some general principles. Dan over here, who's back with us again, is going to give you an overview of general principles in Callset evaluation. I say that because there are much deeper procedures that you can follow for Callset evaluation, but they're very dependent on what you're trying to find out. And so that's why we won't do a deep dive into that. But we'll give you the general principles and at least get you started. Okay, any questions? No, good, so we'll do that. And then it'll be time for coffee break. Yeah?